Hey guys, and welcome back. So the biology paper 2 is coming up, so I decided to do a biology paper 2 walkthrough just to kind of talk you through exactly how I would approach these science questions. Now of course with biology it's a lot of memory work, so the amount that I can teach you is not as much as it is in maths, right? Maths I can show you methods and stuff. However, what I can teach you is exam technique. So exactly how to answer questions and also show you some of the knowledge that you need and hopefully maybe even explain it in certain ways that might help you out, okay? So we're going to approach this in the exact same way we would approach a maths paper, so to speak. So we're going to do it question by question. I'm going to show you the mark scheme answer in as few words as possible because saving time is really important. You only get an hour and 15 minutes. And I'll also kind of explain the science behind it as well, just to add some extra value to this, okay? So let's get on with the first question. With the first question, it shows you the glands in the human body, and you need to, well, first of all, the word gland actually kind of gives this away, okay? But you need to kind of decide which organ system includes these glands. Now, the fact that all of these are glands should hopefully help you in deciphering that this is the endocrine system. And you should hopefully, one other thing to kind of be aware of is that you can't say hormonal system. You have to use scientific words in your science papers, unfortunately. But it is also a good idea to kind of think about, in this question, which glands these are. So you have the pituitary gland, you have your thyroid, you have the adrenal, and you have your pancreas, okay? Now, for men and women, there's actually one more set of glands or organs in this system. So if you're a man, you have your testes, and if you're a woman, you also have ovaries, okay? But again, they cut it off before that part of the image anyway. Part 2, it says which gland produces insulin. Now, insulin is produced in your pancreas, so it's going to be D. This weird kind of blobby chicken leg looking thing is your pancreas, okay? Then it says which gland produces hormones that stimulate the other glands to produce hormones. So essentially, the way I think of this is the gland that controls the other glands in the endocrine system. Now think about it. What part of your body controls the rest of your body? We may have noticed I pointed to my head instinctively. So we know it's a gland that's in your head that controls other glands, right? That should hopefully make sense. Everything in your head controls the rest of your body. So in this question here, it says which gland produces hormones that stimulate the other glands. Well, that means it's a gland that controls the other glands. So it's the one in your head, which is A, which is your pituitary gland. You don't need to know the name, you just need to know it's a pituitary gland. So it makes it a little bit easier, okay? Then it says how do hormones travel from one gland to a different gland? And this is simply through the blood. You can say in the blood as well if you would like. There's no harm in that. You can also say like in plasma or in in certain blood vessels, but the idea is that's just through the blood. Now, this is actually a very interesting part because there's two major systems that control how your body works. You have your nervous system, which is, you know, all your nerves and your brain and your spine, and then you have the endocrine system. So what you might be wondering is what is the difference between your nervous system and the endocrine system? Well, the nervous system is extremely quick because it travels using electricity, right? So it's every time I want to move my hand, right, or do anything like that, it's the nervous system. And look how quick, if I think about moving my hand, I can move it almost instantly. But it's also short term, okay? So it doesn't focus on long scale or large scale or long time changes, right? It's moving your arms and stuff. That's not long term, right? It's a very short movement. The endocrine system is a lot slower, okay? Because your hormones take time to diffuse through your blood, travel around your body and spread out, okay? But it's much more long term, okay? So things like serotonin and dopamine, the things that make you feel happy, right? You generally feel happy for a much longer time than it takes for you to move your arms about, right? Hopefully anyway, or in some people's cases maybe sad, I guess. But also things like testosterone, that's a very long-term effect. For example, ovulation in women, so if you think of the menstrual cycle, that's on a roughly 28-day cycle, so that's a very long-term thing, okay? So that's why, think about what would travel faster, your nerves, which are electricity, very quick, or through the blood, okay? So in terms of the endocrine system, it's through the blood because it's very slow and long-term. Then it says name two glands involved in human reproduction. There's only really two that they accept. And that is the one for men and the one for women. So you have the ovaries. It is also accepted the singular form, so ovary, and also testes for men. And interestingly enough, in the marketing they allow placenta, just in case you wrote that as well, although I don't know, it's fine. Ovulation kits, test kits help women know when they're most fertile. Ovulation test kits detect the increase in the hormone that stimulates ovulation. So which hormone does this? Well, the only one that actually stimulates ovulation in this list is actually LH. Okay, don't forget the other ones are mainly used for blocking the release of eggs and blocking ovulation. So in this case, it is the luteinizing hormone LH. And now we come to our first 6 marker. I love 6 markers in science because there's a misconception which I can correct and help you save time and marks. Some people say that because the quality of written communication is tested, it means you can't write in bullet points. But if you look at the mark scheme, it's in bullet points, right? And all it says, so for the highest level, so 5 to 6 marks, you need to have a judgment strongly linked and logically supported by a sufficient range of correct reasons. So basically, when you evaluate, you say the pros and the cons, right? And all you need to do is have logical points made, okay? So, let's read it. A new contraceptive drug for men is being tested. The drug is given in one injection, it stops sperm from being able to fertilize eggs, and is effective for up to 13 years. So what we're going to do is we're literally going to have a few advantages and a few disadvantages. And in my opinion, because it's 6 marks, it's good to have 3 advantages and 3 disadvantages. Would probably be the best thing to do. However, I'll go through a few others as well. So advantages. Let's think. Well, first one is that it's non-permanent, right? So it only lasts for 13 years or up to 13 years. And the reason why I say non-permanent is, okay, which ones are actually permanent? Well, it's called surgical sterilization, or as you may have heard it be referenced, a vasectomy. Like a vasectomy, which again, you can call it surgical sterilization. Right? Again, think about it. We're talking about men here, contraception for men. There aren't really that many options. You have vasectomies and then you have condoms, right? So it's non-permanent like a vasectomy. It's also longer lasting. So we've compared it to vasectomies. We said, well, it's not permanent, right? Unlike vasectomies. And one other kind of benefit to the condom idea is that it's longer lasting. Okay, it's one injection, and then for over a decade, you won't be able to, get a girl pregnant, I guess. And one other thing is that it's, if you think about this kind of surgical sterilization, look at the word surgery. Think of what a vasectomy is. It's a surgery. Generally speaking, surgeries aren't the best thing because you have to go under anaesthetic, which has its own risks. So it's good because it's just a simple injection. An injection rather than surgery. So the main benefit here is that it's long-lasting like a vasectomy, but it's not permanent and you don't have to undergo surgery. So let's think of some disadvantages now. And literally this is how I'd lay it out, by the way. Right? No harm, no foul. It's logically ordered. They're logical ideas. Well, let's think about it. What's the benefit of having either a vasectomy or a condom over this injection that lasts for up to 13 years? Well, first of all, there's no protection from sexually transmitted diseases. No protection from STDs, unlike condoms. That's one pretty major benefit. There is another thing, it's being tested. So we don't, unknown side effects, right? Effects, as it's in the trial stages. Again, I'm writing in a bit of shorthand, so you'd have to say because it's in trial stages. What else? Well, here's a pretty big problem, is that you can't stop it, right? So think of this situation, right? So, you know, you're in a relationship with someone, you take this injection so you can't get them pregnant, but what if after 6 years you want to have kids? What then, right? Or after 10 years you want to have kids? 13 years is an incredibly long time for something that doesn't seem reversible. Even with a vasectomy, there is a chance of it being reversed or being able to be reversed. With this injection, it just says, for up to 13 years you can't have a baby, right? But that's not necessarily a good thing for people, right? So, for kind of like wearing condoms or anything, you can't kind of undo it midway through. So then, since this is a bit of a vague statement, I'd actually justify and say you can stop wearing condoms, right? If you want a pregnancy. And then obviously if you're looking for a woman's point of view, they can stop taking the pill or using the IUDs or whatever they're using in order to as contraception. They can stop that at any point. You can't, as far as we know, at least from the information we're given, we can't stop this injection. We take it and our sperm is unable to fertilize eggs for 13 years, which is a very long time. I mean, if you're taking this exam, you're probably 16 years old. That's most of your life, right? It's a long time to not be able to have kids, assuming that's potentially what you want in the future. Some other things is, obviously with injection, there's a minor risk of infection. Anytime you have an injection, that can be a problem. You also don't exactly know how long it will last. It says up to 13 years. Does that mean after 5 years there's a chance that it runs out? In which case it's kind of useless, because how am I meant to know? Is there some kind of test that I can do to see if I'm still infertile or not? Again, after 10 years, 12 years, what if it's more than 13 years? So we don't know the actual length of time is another one. It's not as long lasting as a vasectomy. And yeah, unknown efficacy, because it says up to 13 years and it says effective. What does that mean? Is it a reduced chance of fertilization, or is it no chance? They don't tell us. So those are some of the kind of things you can think of, but I would stick to bullet pointing, because as you can see, I have 3 bullet points up here, 3 down here, 6 marks, 6 bullet points. That makes sense. Next question is all about biodiversity. And it says, describe the trends in the money spent on conserving biodiversity from 2005 to 2011. So what I might do in the exam is use my pencil and just like draw a little line here to show that I'm only reading up to that point, and then describe the trends. So as you can see, and literally as you read it to yourself, that's basically what you write. So it increases up to 2008 and then it starts to decrease, which is the exact answer that you can give. So maybe you might make it a bit more fancy. So increases from 2005 to 2008, and then decreases to 2008 to 2011, and that's it. That is completely fine. You don't need to quote figures. However, if you did, you would get marks. Right, so for example, increases to 690 million and then decreases to 630 million. That's completely fine as well. But you can actually just say it increases from 2005 to 2008 and decreases after the fact. Completely fine. Then this is calculate the percentage decrease in the money spent. So change in money spent from 2013 to 2017 over 2013. And then times by 100. Well, this is some maths. 3 marks for doing a percentage change question. It's pretty saucy, pretty nice. So all we want to do is write down how much we had in 2013 and 17. So in 13, that is 690 million. And it's 2017, which is 460 million. So what we're going to do, and you can write working out if you'd like, but you don't have to. It's the change in, so we're going to do 460 minus 690. Again, this is just percentage change, right? Over 690, and then times that by 100 once we have our value. So equals, times 100, a 33.3% decrease. It says just 2 significant figures, so a 33% decrease. That's it. However, if you want to write out the answer, or like the full working out, you just have 460 minus 690. If you're wondering why I haven't written it in millions, so 460 with 6 zeros and 690 with 6 zeros or anything like that, it's because you don't need to, because the millions in the top and the bottom will just cancel out. So, right. Conservation of peat bogs can help maintain biodiversity. Give 2 uses of peat taken from peat bogs. There's a bunch of different things, but the 2 predominant ones that we come across, or at least the ones I like to remember, are just compost and fuel, right? Or burning. But some of the other ones is that it improves soil. So, for example, the drainage or the texture of it. But in general, it just improves soil quality is another one you can use. And that will probably be the main 3. Okay, so compost, don't forget, that's for fertilizing the soil, right? Keeping the nutrients up in it. Describe 2 ways to increase biodiversity in the UK. Well, there's a bunch of different ways. You can do something like plant trees. And it says do not refer to money, so we're not going to talk about money. You can also regenerate habitats. You can also plant trees already done. Reduce pollution. You can have laws that are protecting certain areas, or reduce use of pesticides and herbicides, because those chemicals can damage. Well, they kill things, right? So pesticides kill things like insects and bugs and stuff, which reduces biodiversity. Herbicides kill weeds, which reduce biodiversity as well. Insecticides are for slightly bigger insects like worms and stuff, which again reduces biodiversity. And that is actually it for question 2 for biodiversity. Not too bad of a question in my opinion. The 3 marks on the math I think is quite nice, especially because, you know, we're all maths pros on this channel. This next question is all about classification, which is a lot. It's quite painful for a lot of students, and I don't blame you. It's very technical, and it's a lot of memory, and yeah, it kind of sucks. But in terms of the classification, this first part is always the genus, okay? So it tells you essentially what broad family of organisms it belongs. And Ossifera. And this here is the species name, okay? So, yeah, genus always comes first. It's like you're classifying it going downwards, right, into more precise things. Now it says that it was found from mud. So why was the mud important during the formation of the fossil? Well, if you keep in mind, this is a fungus, right? So it's a very small, very delicate organism, okay? And what's important in the fossilization process, especially when something's formed from mud, is that it doesn't decay. So how do you prevent that decay? Well, it's simple. You stop oxygen from getting to the object, because that means the decomposers can't live on the fungus. Okay? If there is oxygen present, it means that decomposers can start forming around the fungus, and they will start to, well, digest it. They'll start to eat it, in which case there will be no fossil left over. So the reason why there's a fossil is because there's still remnants of the fungus, because there were no decomposers present because there was no oxygen. Okay? It's just a hostile environment for them. The next question. The age of the fossil is in the range of 8.9 times 10 to the 8 and 1.1 times 10 to the 9, so 890 million to 1.1 billion years ago. Calculate the range. All you have to do is take the bigger number minus the smaller number. And I would put it in brackets, because sometimes when it comes to standard form, calculators be a bit weird. They kind of do some stupid stuff sometimes. So I would put them in brackets just to be super duper safe. And that is the range of years. So you can write it, if you want, as 210 million, or you can write it in terms of standard form, which is 2.1 times 10 to the 8. So either 2.1 times 10 to the 8, or 210 and then another 6 zeros. One of those two. Humans did not exist when the fungus was alive. So suggest one other reason why an accurate estimation of when this species of fungus existed is not known. Well, the first thing is that if you look here, there is a range of 210 million years. to put that into perspective, human beings have been around for maybe, modern day humans, well, civilization has been around for about 12,000 years. That's like 200 times that, right? It's a lot. It's a hell of a lot more. It's about what? No, sorry, it's 20,000 times longer than that. So that's one thing, is that this range of values is longer than human existence. So first thing is that the dating is not precise. That's are not precise. Or accurate, you can use either word in this particular case. But you can also say some other things. So for example, one is that, and this is a constant worry for archaeologists, is that we just may not have found a fossil that's older than this. Right? So it says, oh, it lived from 8.9, let's say 1.1 billion years ago. But we could find a fossil in 100 years that's 1.2 billion years old. Or 10 billion years old. The problem is we'll never know when we find the oldest possible fossil, right? So we can never actually pinpoint that. Another thing is that fossils of the fungus could be destroyed, right, over time because of volcanic eruptions or tectonic plates moving. And you can also say that as species evolve, it's very hard to pinpoint exactly when one species ends and the other one begins. Okay, so any one of those things is completely, sensible. Carl Weiss developed the three-domain system of classification. So just for those of you that aren't aware, the three-domain is archaea. Then you have like true bacteria. So you can call them, bacteria if you want. And then lastly we have eukaryota. Okay. Now, archaea are ancient bacteria, like ancient life forms. Basically, they're just all of your extremophiles, okay? But they are truly ancient. Bacteria are bacteria. I know, absolutely shocking. And eukaryota are things like plants, fungi, animals, etc., right? So all of the kind of normal stuff that we think of. So, I just kind of gave away the answer. Fungi are not in this domain. They're actually in the eukaryota domain, okay? And a little bit of a fun fact, but fungi actually have more in common with animals than they do with plants. So, yeah, definitely eukaryota. Now, we're talking about the characteristics, which are features of organisms in the archaea domain. Well, I mentioned that they're extremophiles, and there's a reason for that. So think of all of these extreme things here, okay? They can survive in extreme environments. That's the extremophiles bit. Now let's think of the rest. Well, chloroplasts, I like to use process of elimination, right? Chloroplasts, that's plants or eukaryota, so it's definitely not that. Do not have a cell wall? Maybe, right? Because, again, animals don't have a cell wall either, but plants don't. Can only survive in light? Again, no. If you think about where extremophiles are found, they're actually found near volcanic vents deep under the ocean. So I'm going to say that's a complete no. The cell wall? Maybe. But this thing here is 100% true. So it's those two things. So I like to use process of elimination for questions like this. Carl Linnaeus lived in the 1700s. He classified living things into groups depending on literally their physical appearance. So he looked at them and said what animals look similar, or plants or anything else, which actually is a pretty decent way of doing things, right? Like all dog types look similar to dogs and all cats, etc. But it's not 100% foolproof. Now, what do we use now? Well, one good thing to do, and this is kind of like a bit of a cheat code I used to use when I was in school, is I think about what do we have now that we didn't have in the 1700s? Well, electricity, right, is a one. So we can think about what we have. Well, first of all, studying using electron microscopes, right? So using electron microscopes to look at cell structure. That's a pretty big one. Right. Anything else? Well, chemical analysis is another one that we use. So seeing what kind of proteins are present inside the cells or inside the body, etc. Another thing that they didn't have in the 1700s was DNA analysis, which is a really big one. So there was a big, it's probably before your time, but when I was kind of growing up, they were sequencing the human genome, and they discovered a hell of a lot about kind of DNA analysis of other species as well, like what percentage of our DNA comes from bananas or something weird like that. So this is a really big one. You can also do studies of evolution, so you can trace the kind of evolutionary tree back to see which animals or plants are related to each other. That's another good way of doing it. And also using light microscopes to look at cell structure as well. That is it for the kind of classification question. Not too bad, but there's a lot of stuff to remember, to be honest. Like, again, with biology, I don't think it's intellectually difficult. It's just that you have to store a lot of knowledge in your brain, which I don't consider to be smart per se. I view it as more like you just need to remember a lot of stuff. Okay, and now we come on to my personal favourite biology topic, which is, well, at least for paper 2, which is evolution and genetics. I love this thing because I always thought evolution was pretty cool. So we have a species of bird on a bird feeder, and they use their beaks to reach nuts inside the bird feeder. Cool, fairly standard, absolutely adorable. Let's go. Shows the mean beak length of this species of bird in the UK, and the species of bird often visits bird feeders. So that's a pretty key piece of information there that we're going to use later on. And it says that, so as you can see, there's an upward trend, right? So the beak length is obviously getting bigger. We need to determine the rate of change. So whenever they say that, they just mean find the gradient, okay? Because the gradient is the rate of change. So if we pick 2 points which are cleverly placed to be directly in the middle of 2 points, that looks pretty reasonable. All we're going to do is work out the change in y and the change in x. So if we zoom in nice and deep, that's 13.55 to 13.6. So that's a difference of 0.05. Again, you can use a calculator to confirm that. And what about the year? Well, this is going to be a bit interesting, isn't it? So this goes up in how many squares? There's 1, 2, 3, 4. So it's 5, 10, 15, 20, 25. So in a difference of 10 years, it's 10 over 25. What a weird number to use. All right, fine. So 10 over 25. So every one of them is 0.4 years, and we have 10. So that's 4 years going along, right? So that's 1984. And that, thank God, is 2000. So the actual rate of change is going to be 0.05 over... Actually, we can work out the difference now. That's 16 years in between. 0.05 over 16. Which equals something. 0.05 over 16. 3.125 times 10 to the minus 3. We can do it as a standard form. Minus 3. And then we need some units. It's rate of change, so it's going to be millimeters per year. In fact, it gives it to us in the answer, so I didn't actually have to do that. Anyways, 3 marks for a gradient question. That's not bad, right? I'm hoping you're all thinking that. Part B, or part 2, explain the process of evolution that could cause this trend. Okay, and we need to relate it to the bird feeder at the beginning. So again, just like in our previous part, we're going to have 6 bullet points. But the process of evolution is very, very specific, okay? The first thing you need to talk about is, and this is really a key part, is evolution. It's not a reaction, right? So mutations aren't caused by the change in environment. The change in environment just makes a selection pressure to make that gene better or worse for survival. If it's better for survival, it will get propagated by having kids. So, first of all you want to do is say there is a variation or mutation that causes something. So this works for literally any genetic change, right? So for example, it could be webbed feet. There is a mutation that causes webbed feet. There is a variation, in this case, in beak length. Caused by a mutation. Okay? This is so incredibly vital that you write this, okay? Because again, the variation is not caused by the change in environment. It's caused by random genetic mutation. Okay? So again, this could be anything you'd like. So it could be, if it's like webbed feet, it could be eye colour, it could be size of heads or whatever. But you have to say, for 1 mark, there's a variation caused by a mutation, okay? And that is vitally important, okay? Boom, 1 mark, nice. So, you can also say that the beak length is controlled by genes, okay? Because what you want to do later on is imply that it's inherited to the children. So the variation in beak length, or beak length, is controlled by genes. Now the next few points, we're going to be quite specific about it, okay? Now how does this help? If you look at the bird feeder, if you look at their little stubby little noses, those little beaks, if you look at the middle of the bird feeder, it doesn't look like the birds are actually going to be able to reach all the way to the middle. So the ones with longer beaks can reach more nuts. We're going to state that. Birds with longer beaks can reach more nuts or food. Okay? Cool. Therefore have more energy. More energy. More energy. And offspring. Full marks. Again, I'm telling you, this is literally the most concise way to write it. So again, the idea is that they're getting more nuts, which means they have more energy, which means they're able to have more kids, right? Because they're not starving to death. Okay. When they have offspring, the genes for the longer beak, so genes for the longer beak, are passed on to offspring. And lastly, and this last point is so critical, because to get a level 3, which is 5 or 6 marks, you have to say this when it comes to evolution, okay? You have to say that it's repeated over many generations. It must be linked to several generations, okay? So again, however you want to say this, so repeated over many generations increases mean beak length. So I know it's very tempting when you're kind of going through these 6 markers, right, is to literally write down every single bullet point in the mark scheme. But here's a few problems. One is that's more stuff to remember. In the mark scheme, there are 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 points. You only need 6. So why memorize almost double the number of points? The second thing is that, for example, this last bit over here, repeats over many generations, increases mean beak length. That's how you get the level 3. So let's say you're like, okay, you're right. Why would I do 11? I'm just going to memorize the first 6 bullet points. You can't get 6 marks. Because the thing they're trying to test is that evolution takes place over a very long time. An incredibly long time. If you look here, from 1980, whatever, 82 to 2008, there's only a difference of 0.05 millimeters from here to here. That's 20 years, well, 16 years, 0.05 millimeters. That's 50 millionths of a meter. Right? It's absolutely tiny. Right? And if you look here, it's a 3,000th of a millimeter every single year. So for it to make a noticeable difference, there's probably going to have to be 1,000 years in the future. Because that's 3 millimeters difference. 1,000 years just for a visible difference, right? It's very, very long term, okay? So again, I'm not saying that, don't read the mark schemes and don't memorize them. Memorize mark schemes for sure, but pick 6 points and read the mark scheme to make sure that there isn't one point that you have to say to get the full 6 marks. So I fully encourage you reading the mark schemes, but actually read them. Okay, it tells you everything you need to know. It literally says, for level 3, detail of process of evolution must be linked to beak length and implication of several generations is required. So you can even say repeated over many generations. That's it. Okay, but make sure you actually read mark schemes. Okay. Birds of this species live for about 3 years and have 24 eggs every single year. Explain why evolution is easier to study in this species of bird than in humans. Well, what did I just say? Evolution is only visible over many generations. Human beings live, on average, I think the life expectancy now is 77 years, at least in the UK. So you're going to have to wait for 10 generations of human. So roughly 770 years in order to get any kind of measurable outcome. Right? You'd be a skeleton, right? So the people that start the study will die at the same time the people they're studying will. Right? It doesn't make any sense. So the reason being is A, there's a far shorter lifespan. Okay? So you can say here, more generations in a shorter amount of time. There's also more offspring. 24 eggs every year is a lot. The average number of kids that people have in a couple is under 2 in the UK. Right? Whereas now we have 24 every year. So more offspring. So those are 2 points already. So what does that actually mean? And remember, we're thinking of evolution here. Okay? So far, we've stated 2 facts. Shorter lifespan, more offspring. You can get that just from reading the question. So explain why evolution is easier to study in this species of bird than in humans. Well, think of the difference. Human beings live longer than 3 years, so this must be some kind of benefit. So you don't even need to know anything about science. You can just say, well, shorter lifespan. Because it's the only difference they've given you. I don't know any human being that's had 24 kids in a year. Right? So more offspring. Those 2 things you can get without any knowledge of biology, right? Well, assuming that you don't think human people can have 24 kids a year or something, or they only live for 3 years. So the third mark, and this is the grade 8, 9 stuff, is saying, why is this good? Well, again, remember that in order for us to see any difference, it needs to be repeated many, many times. So if you have more generations and a shorter lifespan, it means the genetics change faster. Right, so the genetics of the population something that might kind of interest you in terms of this, right, is that human beings are found to have been growing taller over the years, right? However, the actual speed is ridiculously slow. The average height hundreds of years ago is the same as the average height now. So it doesn't really change, right? So keep that in mind when it comes to genetics. Although like humans are evolving and everything, it's taking thousands of years, right? It's a very long, lengthy process. Birds of this species are found in different parts of the world. Describe evidence that would show 2 individual birds are the same species. So the 2 things you want to compare when you're talking about species is something called phenotype, which is how something looks. So you can say similar or same phenotype. So again, that's how something looks. You know, if you go across the wall and you see a 4-legged cute little animal that loves like licking things, it's probably a dog, right? And you're talking about genotype, which is the genetics. So all you have to do is take some cells and have a look at them, read the DNA, do some DNA analysis. Now the last thing is the actual definition of a species. Okay? The definition of a species is kind of, you know, animals that have, or I'm going to assume animals, but you can technically have plants as well, the similar phenotype, similar genotype, and they can produce fertile offspring. Fertile being the key word. So it can produce So for example, if I get a dog from the UK and I fly over to China and I see another animal that looks the same, I take some cells and I compare the DNA, the genotypes, and they're similar, and I can breed them and produce fertile offspring, then they are the same species, but only if they can produce the same offspring. So you may have heard of things like ligers, which are tigers and lions that are bred together. So they can breed and produce offspring called a liger, but they are not the same species because they are not fertile. You can't breed two ligers together. Okay. Same with it's not mules, but it's like donkeys and horses you can breed, but they're not fertile offspring, which means they're different species from each other. Okay. So that's how you can test. Lions and tigers are not the same species because their offspring are not fertile. So that's a very key part down here as well. And we're continuing with some more biology. So for question 5, we have some reaction time. So caffeine is a drug that decreases reaction time, so it makes your reactions quicker. A group of 16 students investigated the effect of caffeine on reaction time. The students were all 15-year-old girls, and they were divided into 8 pairs of students. This is the method used. So we have student A starts 2 stopwatches at the same time, hands one over to the other student. Student A then says stop at the same time as pressing the stop button on the stopwatch, and student B will stop the stopwatch the second they react to it. And the difference in time shown is recorded, which is the reaction time of student B. Student B drinks a caffeinated drink, and then they redo it and they see what the difference is. So essentially, if you imagine it, on student A's, maybe it's going to say 1.50 seconds. And on student B's, it might say something like 1.54 seconds, which means the reaction time is 0.04 seconds in that particular instance that I've demonstrated, okay? So the idea is that, again, student A clicks both of them, hands one over, then goes stop, and then boom, clicks the other one. The difference in that is the reaction time, as shown here. So that's just an example. So that's what I like to do whenever I come across any kind of experiment in science, is I think about it. And I imagine myself doing it, and I think about what could go wrong. I think about what exactly is happening. Does it logically make sense? I can see why they'd think this would be reaction time, so it's all good. Now, it says here, suggest one control variable the student should have used in the investigation, and there's like a billion, right? But it does say do not refer to age or sex because it tells you 15-year-old girls. So you can't say male or female or the age because that's already a control variable, okay? So it could be anything, right? So think about what else would affect reaction speed, okay? So again, they're trying to measure reaction speed. So think about what could affect reaction speed. Well, think about any time that you felt a bit sluggish or felt particularly on edge. Well, one thing that I would think of is that you need to test the amount of sleep, right? So keep in mind, it's not just 2 students doing it, it's 16 students doing it. So if some of them have got 3 hours sleep last night, they might be sluggish, their reaction time's slower, right? Whereas someone that's well rested will have a faster reaction time. The other thing that you need to take it, so some other things that might need to be taken into account is things like body mass. If someone is heavier and bigger, they need more caffeine to have the same effect on their reaction time as a smaller person. So if you gave a little child some caffeine, they'll be hopped up and shaking and sprinting around. If you gave a full-grown adult a cup of coffee, it's not going to have that much of an impact on them. Okay, so just have a think about what else would affect the thing they're trying to measure. Okay, so in this case, reaction time. What else would affect it? Cool. Part B. Suggest 2 sources of random error when using this method to measure a person's reaction speed. So various things can take place. So you can have like a malfunction with the stopwatch. Maybe the stopwatch doesn't work properly. Sometimes, especially if you use the ones in like PE, sometimes the button's all a bit weird, so it doesn't actually start and stop when you press it. You could have that student B is distracted. So B is distracted. Student A not pressing start at the same time. Not starting stopwatch. Starting both stopwatches at the same time. At the same time. And another thing is that, when they say stop, they might not press stop at the same time they say stop. They might go stop and then press stop, press. Slight difference there. Just think about all the different things that could go wrong in terms of measuring reaction time. So the stopwatch not working, student B not paying attention, right? If you've done experiments in school, you will know that, young people get distracted sometimes. Your partner might not be paying the most attention. Another thing, especially with testing reaction speed, is that people kind of show off a bit. They want to have a fast reaction speed, so they might anticipate you saying stop. So if student A is going, then the other person might just press stop straight away, in which case you have a negative reaction speed or something like that. Okay, so the idea of like student B trying to anticipate when student A is going to say stop, so maybe you blindfold them to prevent them from kind of anticipating it or something along those lines, but those would be some sources of random error. All of them are completely fine. Here we have a bit of a statistics question, but you have all these different times here in a table, and it says, why can a mode not be determined? Well, think about what the mode is. It is the most common value. So basically, you find out which number has come up the most number of times. If I look at these values, I notice something. None of them are repeated, which means there's no mode, because none of them turn up the most frequent, right? They're all equally frequent. So simply put, there are no repeated results. So you might want to pick a mean instead of a mode. Students decide the result from pair 8 was anomalous. The students calculated that the mean decrease in reaction time was 0.03 seconds. Describe how they calculated the mean decrease in reaction time. Okay, so first of all, pair 8, it makes sense that that's anomalous, right? Because it's well out there. The next highest one is 0.041. They got 0.097. Okay, so unless that person was literally asleep, it's unlikely that that's an accurate result. So how would they calculate the mean to get 0.03? And you can test your answer if it's correct, because if you write your instructions and you follow them and you don't get 0.03, you're not right. So here's what you do. How do you calculate the mean? You add up all of the values and you divide them by the number of values. That's all you have to write. So add up the other 7 results, because remember, they're getting rid of the number 8, right? Other 7 results, and then divide by 7. Remember, when you take a result as anomalous, you effectively delete it. So it's as if it doesn't even exist. So you're only treating these other 7 answers over here, okay? So don't include them in your mean calculations, or any calculations, in fact. If it's anomalous, it means it's not correct. So, caffeine causes the release of adrenaline, and adrenaline affects heart rate. It increases heart rate. So we need to explain how the effect of adrenaline on the heart rate might cause reaction time to decrease. Well, the first things first is that it increases heart rate. Again, if you ever had a lot of caffeine, you would experience that your heart starts beating really quickly, increases heart rate. Or if you've ever done something exciting, you might feel your heart beat really, really quickly because of adrenaline, like if someone scares you, anything like that, on a roller coaster as well. So now, when I approach these questions where it says explain how the effect, I need to take this in steps, right? So imagine you're trying to explain it to someone. So I'm a teacher, right? So when I try and explain this to you, if I said, caffeine increases reaction speed, and you ask me why, how would I explain those steps? Well, adrenaline increases heart rate. What's the effect of that? Well, it means that you're going to have more oxygen and glucose, right, passing around your body. Increases glucose and oxygen. And what causes reaction time? Well, it's your brain. So to your brain, but not just your brain, also your muscle cells, right? Because it needs to go to your brain and your muscles. To brain and muscles. Okay, and what's the effect of that? Well, it increases the rate of respiration. If your heart starts beating faster, your breathing's going to increase, and therefore your respiration in your cells is going to increase because there's an abundance of the ingredients required for respiration. So increases rate of respiration. Okay, and what does that mean? What does increasing the rate of respiration actually cause? Again, remember, respiration is not breathing, that's inhalation and exhalation. Respiration is the process by which energy is released from glucose. Okay, so you're going to have more energy. And what does that mean? More energy means that your muscles can contract faster. So, more energy released causing muscles to contract faster. And this is a genuine thing where people try and actually increase their adrenaline before doing some kind of sporting event because it makes their muscles more efficient. So, for example, on sports day, if you ever get nervous for sports day, your heart starts beating, you will run faster and further than you've ever done before because more energy has been released in your muscle cells. You don't have to build up to it, you just have that energy straight away. So again, anytime you have an explain question, imagine you're trying to explain it to your younger sibling, right? I don't know why you would be, but just pretend you are. Okay, what you would do is you take them through a series of steps which lead them to the actual answer. So why does caffeine increase or decrease reaction time? Well, it increases your heart rate, glucose and oxygen passing around your body, which means it goes more to your brain and your muscles. That means there's more respiration, which means there's more energy, which means your muscles can contract faster, your brain can react quicker. Okay, it's a very set series of steps that occur. Adenosine is basically the opposite of adrenaline. It's basically the opposite of caffeine in that sense as well. It's a different chemical made by the body. It binds the receptors on the relay neurons and it decreases the number of impulses. So what that effectively does is it decreases reaction time. Increases reaction time, but it decreases reaction speed because the messages aren't passing through your neurons as quickly. Okay? Figure 5 shows how caffeine binds to the adenosine receptors on a relay neuron. When caffeine binds to the adenosine receptors, it blocks the receptor so adenosine cannot bind. So here you have caffeine, as you can see, it blocks adenosine from fitting into that gap, which means the messages aren't being slowed down. Okay? The messages can continue passing as quickly as normal. Label X shows the gap between the sensory neuron and the relay neuron. What is the name of this? So this kind of intermediate area here is something called a synapse. You can also call it the synaptic cleft. Oh god, I can't spell it. Synaptic cleft if you're feeling particularly fancy, but the synapse is completely fine for that case. And 5.7 says just suggest why the reaction time decreases when caffeine binds to the adenosine receptors. Well, it's as I just said. Now, they do like to do this where they introduce chemicals you may have never heard of before, but they explain what they do. Adenosine decreases the number of impulses in relay neurons. Okay? So if caffeine is preventing them from binding, then it can't decrease the number of impulses. That's how it causes reaction time to decrease. Okay? So caffeine prevents adenosine from binding, which means fewer impulses are blocked. So impulses are blocked less. So the impulses are more frequent. Okay? So very simple, very short sentences. You don't need to do too much explaining. You're more than welcome to, but you don't need to, right? It's just very simply following the chain of logic they've given you. So adenosine binds to the receptors, which decreases the impulses. Caffeine stops that. Okay, but that means fewer adenosine bind, which means the impulses aren't blocked as much, which means they're firing more frequently. That's it. Okay, so it's just very much following the logic in the question. And lastly, we have once again some more genetics and mutations and stuff, which I get is probably one of the harder topics in Paper 2 biology, but hopefully the way I explain it will make a bit more sense. So it's about genetic disorders, which I also find quite interesting. Some people are heterozygous for a genetic disorder. To find the term heterozygous, well, all you have to remember is that there's two types. There's homozygous and heterozygous. And I like to remember it as homo being same. So homozygous means that you have two of the same alleles. Okay? They're identical. So hetero must mean the opposite. Means you have two different alleles for the same gene. Okay? So specifically, that means we're going to have a recessive and a dominant allele, okay? Because, again, if you're heterozygous, it means you have one of each. So, again, homo being same. Homozygous means they have the same allele, so maybe both dominant or both recessive. And hetero just means the opposite. It means they have two different alleles. That's what we have to think about. Now we have a 6 marker based on determining the probability that a child will be male who has the disorder. I mean, you have to use a Punnett square, which is somewhat fun. So if we have a look at this, okay, we have this kind of family tree, and it says that white circles are females without the disorder. Now bear in mind that even if you don't have the disorder, you can still carry the genes for it if it's recessive, okay? Squares are males that don't have the disorder, circles are females with the disorder, and black squares are males with the disorder. And it says person 7 and 8, which are these two people, are planning to have a third child. Determine the probability that they're male, so we should highlight that, who has the disorder, okay? And you should draw a Punnett square diagram, identify the genotype of person 7 and 8, identify the phenotype of each offspring genotype, and use these symbols H for dominant, H and lowercase h for recessive. Okay, so first things first, if I have a look at this, and I follow this along, I know for a fact that this genetic disorder is recessive. How do I know that? Well, both of these people do not have the disorder, and yet they had 2 kids with the disorder. So what that means is that they must carry the recessive allele. So wait, let me just check. Yeah, they must have the recessive allele, right? So I know for a fact that this must be a recessive trait, okay? Because, again, they carry it, but they're not displaying it. So that means both of these people, so for number 7, must have the recessive allele, okay? So I would write this out just to make it super clear. So person 7 is 2 lowercase h's because they display the recessive trait, okay? So hopefully that makes sense. First, you determine if it's a recessive trait or not. Here's a small hint. Most genetic disorders are recessive, but you can check. If 2 people that don't show a disease or a trait or a characteristic have kids that have the trait, then it must be recessive, okay? And this is in the same way of, like, for example, people with blue eyes, right? You can have 2 people that don't have blue eyes have a blue-eyed kid because they may both have the genes for blue eyes. It's just that blue eyes are recessive. Cool. What about person 8? Well, we don't know much about them, but we do know that they've had a child that has the disorder. Now, the thing about recessive traits is that you must have both parents carrying the gene, okay? So a recessive disorder has to have both parents carrying the gene. Okay? Because think about it, if this person's lowercase and this person's HH, their kids can only ever be, at best, 1 lowercase, 1 capital, which means they would not display the characteristic. Okay? So if that makes sense. This person cannot have both dominant alleles because they have a kid that has the disorder. Okay? So again, if you were to have a blue-eyed person and let's say a brown-eyed person, if that brown-eyed person does not have the genes for blue-eyed kids, they will physically be incapable of having blue-eyed kids. Okay? So you'd have to look at their parents and see what kind of genes they have. So because we know that this kid down here has 2 lowercase h's, the mother must carry at least 1, or in this case exactly 1, recessive allele. Okay? Does it matter which way around the h's go? Absolutely not. Of course it doesn't. Okay? You can have his h, lowercase h, or lowercase h, capital H. So I hope that makes sense, okay, for kind of determining where that genetics comes from. Okay? It's kind of like a logical puzzle in that sense. So again, one last time, if a child has the recessive trait, both parents have to have, at minimum, 1 recessive gene each. Okay? Just like here, they both had to have 1 each to have kids that had the recessive trait. Okay, now we need to draw a Punnett square diagram. Now how does this work? Well, all you have to do is write person 7 and person 8. And then people draw them kind of differently, but that's completely fine. And then what you're going to do is you're going to show both possibilities. So person 7 is a HH, and person 8 is a H, lowercase h. Again, it doesn't matter the order. You're going to get the same result afterwards. So if I drag that down a bit, now we can actually draw the square itself. So I mean, don't you just love how artistic I am? Like, it's amazing, truly. Again, I'd probably draw this with a bit of a ruler, but anyways, let's get rid of those 2 lines. Okay, perfect. And something along those lines. So now all you do is you match them together. It's just like a multiplication table. So H, H, so that's H, 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 then H, H. It's literally a multiplication table, right? You're just matching them all up. Nice and easy. So, by the way, the actual drawing of the Punnett square, even though mine looks horrific, it's completely fine, right? It doesn't actually matter as long as you get the right thing. So, as you can see, 2 out of the 4 kids will have the disorder, right? So, 2 out of 4, which is the same as 0.5 if you put it in your calculator, have the disorder. That's the probability of having a kid with this trait. But that's not the answer, because then it says the probability that they'll be male. Okay? Now, how many kids are male? Well, it's going to be half of that. So then you're going to divide this by 2, so 0.25 for male with disorder. Okay? So again, 0.5 for just with the disorder, 0.25 for male with the disorder. Okay, because remember, half of kids are male, half of them are female, at least in terms of this particular kind of question and GCSE science. And the last question of the entire paper shows a different disorder or a different inherited trait. So polydactyl is a different inherited disorder. 2 parents did not have any alleles in their ordinary body cells, but they produced a child with polydactyl. Explain how this suddenly occurred in this family. So what they're trying to get at here is how do these disorders come to exist, okay? And the way they come to exist is through random mutation. So the first step is literally caused by mutation, which is how a lot of traits nowadays form. Like different hair colours and different eye colours actually form from random genetic mutations. Now we have to say it's saying explain it, so caused by mutation, okay, but how does the mutation happen? Well, it's caused during the process of meiosis, when the cells are splitting, and what happens is the wrong amino acid sequence is put in. So the amino acids are put into a different sequence, which causes a different expression of the gene. So, caused by mutation, during meiosis, so specifically that's the production of germ cells, right? So sperm and egg cells, which leads to a different sequence of amino acids. And what does that actually mean? It means different proteins are produced. Okay, so you can either say different proteins are produced or certain proteins aren't produced. Both of them are technically the same, right? In that case, the idea is that something different happens. So again, during meiosis, when the amino acids are being sequenced, right? So when like RNA and tRNA is going along, etc., 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 the order can sometimes be changed just by a mutation, by random luck. Okay, so something goes wrong, and what that leads is to a different protein is produced, which leads to something happening. Most of the time, it's a negative trait, right? So a lot of the time, you might be born without the ability to produce insulin or something like that. So most of the time, it's actually a negative trait, but then sometimes again, you have something normal like eye color, but then also things like being able to walk on 2 feet, right, with a straight back and things like that, and opposable thumbs. So all of those things can be produced by random mutation, because that's exactly what happened with human beings. But that brings us to the end of the paper. I hope you found that somewhat useful. I'm not too sure about a biology paper because a lot of it is just memory. So hopefully I showed you some exam technique in terms of answering the longer questions. But the chemistry and physics papers, especially the physics, I think will be a lot more useful in that sense because there's a lot more calculations and problem solving involved. But other than that, I hope your revision goes well and hope the exams are going well for you.